produce pancreatic juice. They secrete pancreatic juice, which is the digestive juice. It's going to travel through these ducts and get into the small intestine and mix with the chyme at that point. So again, it's not equally distributed at all. I mean, 1% and 99%. But we're going to talk a little bit about this pancreatic juice because it's so important. Each day with the pancreatic juice, you and I will secrete, I'm talking about pancreatic juice now, we secrete somewhere around 1.2 to maybe 1.5 liters of pancreatic juice per day. So most of you are familiar with the liter bottle of seltzer that you can buy. So figure about 1.5 liters of that's quite a bit of liquid that you're secreting out of the pancreas each day. The pancreatic juice, if we take a look at it, it's water-based, so obviously we're going to have water in it, but it also has quite a large amount of something that's referred to as sodium bicarbonate. Major component of the pancreatic juice, water and sodium bicarbonate. This is not a chemistry class. I'm not asking you to memorize this, but that's the chemical formula for sodium bicarbonate. Very, very important. These components are extremely important. The chyme is coming out of the stomach at a small amount, maybe about a tablespoon, I should say, per pulse of the stomach. And then we have to get everything under control. You think about the chyme coming out of the stomach. What's the pH of the chyme coming out of the stomach? What's the stomach's pH? Yeah, 1 to 2. It's very, very acidic, this chyme coming out. So we have the chyme coming out of the stomach, and as the chyme comes out, the chyme has hydrochloric acid mixed in with it. The small intestine is not designed to handle it. It doesn't have the mucus coating like the stomach, and you can cause major problems. Well, we mix it with this sodium bicarbonate. We take the sodium bicarbonate, and in the liquid of the pancreatic juice, it mixes with the hydrochloric acid, and then we have something that's referred to as an exchange reaction take place. We exchange component parts off of this. So the chlorine over here comes off of this and combines with the sodium. So we produce sodium chloride, which you know more commonly as table salt, plain old table salt, not a big deal at all. That leaves us with a hydrogen ion over here and this bicarbonate portion over here. So we end up over here with something H2CO3. So we have salt over here, salt. This is something that's called carbonic acid. Carbonic acid is a milder acid. It's not as strong as hydrochloric acid. It's much milder. I know it's a rainy, boring day, and I'm already putting you to sleep. I apologize. But if you take a look at this carbonic acid, there's two things that we can break this down to. We can decompose it into something that won't bother the body. If anybody sees any components of that. Carbon dioxide? Yeah. Okay, carbon dioxide we can get out of this. So we took that out of that. What does that leave us with? Right, we got two H's and we have a leftover O. What? These two O. So we're able to take this chyme that will damage the inside of the small intestine, and we're able to neutralize it. We get some salt, we get some carbon dioxide and water, not a big deal. The body can easily handle it. We don't damage our small intestine at that point. So we certainly find that in pancreatic juice. We're also going to find in the pancreatic juice what I like to refer to as a cocktail of digestive enzymes. So the cocktail of digestive enzymes is found in the pancreatic juice. We have enzymes for digesting everything. Now, obviously, if carbohydrate digestion stopped in the stomach because it was so acidic, it destroyed the salivary amylase, you can imagine what happens to the pepsin in the chyme once we neutralize the chyme. The pepsin doesn't work anymore at that point. It needed to be in an acid environment. Well, we have this cocktail of digestive enzymes. I'm not testing you on the name of these. I just like to give you the whole picture, what we're releasing out here. There's an enzyme called trypsin, and another enzyme called chymotrypsin. There's something called carboxypeptidase. and another enzyme called elastase. These are all for different protein components. You stopped the pepsin from working over here because we neutralized it. Well, now I have to add in some new digestive enzymes for protein digestion. And so we're going to have these enzymes that are released out. There's also some enzymes. One is referred to as deoxyribonuclease, and another one that's called ribonuclease. So we have deoxyribonuclease and ribonuclease. It's going to digest, as you can tell by the names, DNA and RNA. We're eating plant cells, we're eating animal cells, there's DNA and RNA in it, and we can break them apart and digest that. So those are two other enzymes. We're also going to now introduce something called pancreatic amylase. We had salivary amylase, but that stopped working in the stomach. Now I got to continue my starch digestion, my carbohydrate digestion. I'm going to add in here pancreatic amylase, so this would deal with the starches still that are in the food. This over here deals with DNA and RNA. And then lastly, we're going to really give a large hit to this now of something called pancreatic lipase. And that would be for the fats, obviously. It's a cocktail of digestive enzymes. We have digestive enzymes for digesting everything: proteins, carbs, lipids, and even DNA and RNA, all found in there. So when the food leaves the stomach, message goes from the small intestine, hey, food's entering here, goes up to the brain sends a message down to the pancreas, says, dump out some pancreatic juice. We need to neutralize the acidic kind, and I want to get some new digestive enzymes going here so we can continue digestion. So that's what's going on with the pancreas. And then we have another big organ over here that gets into play. That's the liver. Let's talk a little bit about the liver. We won't get through it today, but we'll discuss some of the liver. The liver is the largest internal organ of our body. It's about 3.5 pounds in size, so it takes up a lot of room. It takes up almost the entire upper right quadrant area. So it's about 3.5 pounds in size. It's completely covered with peritoneum on the outside of it, held in place by something called the falciform ligament. I spoke about that the first day we were together. So you can see the liver here. You have the falciform ligament that runs in the middle and then attaches to the diaphragm. It kind of holds it in place there. So we have, 3.5 pounds. It's divided into two lobes. There's two lobes to the liver, a right and left lobe, and it's divided by the falciform ligament itself. That's what separates those two lobes. If you take a look at the picture above me, we have a right lobe, which is a little bit, this is a larger lobe, and then you have the left lobe that's a little bit smaller. So it has two lobes. Now, that being said, I gotta mention something here. Again, not for my test, but for a test that you may be taking along the line. There's only two lobes to the liver. There's a right and left lobe to the liver separated by the falciform ligament. Whoever writes a lot of these entrance tests, I swear, don't know any anatomy or physiology, because I've seen questions in the books that you buy that you're studying out of. All the books swear that there's 4 lobes to the liver. There's 2 lobes to the liver. There's a right and a left lobe to the liver. Now, the right lobe is so darn big that right lobe is a big lobe, as I said, and it has little extensions. Off of the right lobe, we have something that's referred to as the caudate lobe, but it's part of the right lobe. And there's another one called the quadrate lobe that also comes off the right lobe. But they're not lobe, they're part of the right lobe of the liver. But if it shows up on your HESI test or your Keys test, and it says, how many lobes does a liver have? Give them 4. They're wrong, but that's the answer that they're gonna want. And you'll find a lot, even in your allied health programs when you get there. I had a friend that went through the nursing program here years ago. She lives in Florida with a husband now, but when she was in the program, we used to go over, so we'd have coffee, go over stuff. She wanted to know more. She just didn't want to know the facts. She wanted to understand why. What's the physiology of why this is happening? I'd read half of her notes, and they were all PowerPoint crap. I'd read them and say, that's wrong. That's totally wrong. You don't get lower sodium with this disease. You get higher sodium, and it affects the calcium. And they had it wrong. I said, just give them what they want. They're wrong, they're totally wrong. So if they ask how many loaves, give them 4. Doesn't matter. It doesn't matter. Just get into your program and then forget about it at that point. The loaves themselves now are divided into still smaller segments called lobules. Kind of similar to when you learned the respiratory system. You had the lungs. Remember the lung? We have lungs, things called lungs. 3 lobes on the right side, 2 lobes on the left side. But the lungs are divided into smaller segments called lobules. We have these little lobules. And each lobule, the lobules have 5 parts. There's 5 different parts to a lobule. I'm going to go over them, but I liken it to when you were learning about an osteon, if that name sounds at all familiar from Anatomy 1 within the bone. You had osteon, little microscopic units. There were 5 parts: a central canal, concentric lamellae, lacunae, and osteocytes, canaliculi. There's 5 different parts to each lobule. And we have quite a number of lobules. There's about 100,000 lobules that you have in an average liver. So it's not just a couple of lobules, there's 100,000 of these things. But each of the lobules has 5 component parts. So let's just take a look at these 5 parts briefly. I'll put up a picture. Each lobule has in the middle of it something called a central vein. The central vein is draining the blood from the lobule. Veins drain the blood, so you're draining the blood that's in the lobule. So they all have a central vein. They all have just rows of hepatocytes, liver cells. There's these rows of hepatocytes or liver cells, and they radiate out around the central vein. So they're going to radiate out all around the central vein. Sort of like the spokes on a bicycle wheel radiate. I'm going to put a picture up so you can see it. Between the rows of hepatocytes, we have something that's called sinusoids. Sinusoids, think of them as capillaries. We'll just say capillaries. If a central vein is draining blood, it had to come from somewhere. It's traveling into sinusoids. They're not really capillaries. Otherwise we'd call them capillaries. The sinusoid's a little bit bigger than a capillary, but smaller than a arteriole. But it's a capillary for all intent purposes. We also have lining these sinusoids a cell. It's the longest cell name in the whole body, and if you get it in a Scrabble game, you win. You're going to win with it. You're not going to miss this. It's something they refer to as stellate reticuloendothelial cells. Stellate reticuloendothelial cells. The stellate reticuloendothelial cells actually line the sinusoids. And I think that's a big name with that. I didn't think anything was in the body with a name that long. The term stellate just means star-shaped, like the word stellar. When you think of stellar, it's a star-shaped appearance. And then reticuloendothelial cells, the endothelium is the lining of a capillary. It's the lining of the inside of blood vessels. They adhere to the inside of them. Actually what it is, it's got a big fancy name. Years ago they were called Kupffer cells that didn't tell us anything. Well, what they actually are, they're phagocytes. Phagocytosis, Anatomy 1, Chapter 3, cell eating. Think of these guys as stationary Pac-Man. Instead of them moving around eating stuff, they sit there and wait for the blood to come by and they eat stuff. You probably at one time or another heard that the liver is one of the organs that cleans the blood of the body. That's what it's doing, cleaning up the blood. And then the last feature that we have associated with it are little structures called biocanaliculi. And then I'll show you a picture we'll go over. We're going to end there. I'm not going to give any more notes at that point. I don't want to get into the blood supply and stuff. We'll take a look at it. I'll let you copy that. Sorry for such a boring discussion. It's not overly exciting. Plus, it's a dreary day. And hot in here. It's hot in here? Okay, yeah. I'm still complaining. I'm going to get them. I got them now. I spoke to the 2 people that were in charge upstairs. I can tell you, the office is Professor Flannery's office the other day was 86 degrees. It's off the chart. Yeah, it's terrible. Our office was 82 yesterday. So I've spoken to the 2 people that were in charge of things. Nothing happened. So now I called the academic vice president. I'm going to see what happens. But I actually don't know. I'm going to go down. I made an appointment. I found out that the ocean